Okay, so let's talk about this apparently terrifying concept of osmosis, and let's see if we can simplify it so that we all have a really good understanding. We've got the definition there, but I want to come back to that actually in a few moments' time. The most important thing I could say to you at this specific stage is if you haven't watched the tutorial on diffusion, you need to do it. I actually recorded it a few moments ago and it's just up here. I really encourage you to go and have a look at that diffusion tutorial because I'm going to be relying on your conceptual understanding of that as I go through this tutorial on osmosis. So I can't really stress that enough. I think it's important for you to do exactly that. The second point I would make before we kind of really get into the nitty-gritty of this is I think it's really important to realize, and I'll actually write this in fully for you, okay? So just bear with me and I'll talk you through this as well. So I want to make a point that the concentration of solutes, remember a solute is within a solution is the smaller, that which there's less of. The concentration of solutes, and we could be talking about things like minerals, we could be talking about things like sugars, for example, but their concentration inside cells needs to be stable, okay? Generally speaking, needs to be stable on the whole, okay? For good functioning, all right? So for cells to do their jobs, broadly speaking, we need to understand that the sugars and the minerals, amongst other things, within those cells need to be at relatively stable levels. Therefore, we need a methodology that enables that to be the case. But at the same time, let me just write this in green here, but, and that's a really important but in this case, the solvent, and of course the solvent when we're talking about cells in most functions within plants and animals, we're talking about water, but the solvent, that which there's more of within a solution, can vary greatly, okay? So we need to understand that because that kind of seems somewhat counterintuitive when we think about the process of diffusion in isolation. It can vary greatly and can pass through the membrane. So we want to look at the situation where we're looking at two sides of a membrane through the membrane. We're looking at two sides of the membrane. It may well be that things like salts, sugars, for example, don't pass. Let me restate that. Minerals and sugars don't pass through the membrane, but water continues to do so through the process of diffusion. And we have a name for this, which is called osmosis. And we see it here. Osmosis is the diffusion of water from a dilute, okay, that where there's a lower concentration of, let's say, the sugar, to a more concentrated solution through a partially permeable membrane that allows water molecules through. So that's fine. We can define it, and that's sort of helpful. But let's see if we can kind of picture it. All right. So what I'm going to do here is I'm going to draw a dotted line. But in essence, let me draw these little dashes a little bit longer. These dashes. This is not just a line. Okay. We are going to assume that this line represents what we described a moment ago, which was a partially permeable membrane. Okay, so let's assume it's something like let's just loosely call it an animal cell for this time. So let's not be specific. Let's just loosely call it a eukaryotic animal cell. Okay, and what we're going to find here is that on this left-hand side we have, I don't know why I've done this in orange to be perfectly honest, but we have a load of water molecules, okay? The colors don't mean anything at this point, okay? So on this left-hand side, effectively, let's call this outside of the cell, we have a whole bunch of water molecules, okay? So we can first of all refer to this as what we refer to as a dilute solution. You'll see what we mean by that in a few moments' time. Now, on this inside of the cell, over here, we've also got a bunch of water molecules, okay? But within this, we've also got some big sugar molecules. So let me carry on. We've got some water, but we've also got some sugar molecules. Okay, I think I've done a smaller one for some reason there, but you get the idea. It's still much bigger than the water. So we've got another sugar here. Make that one a little bit bigger. And let me do another one down there. I think I did a different color this time, different blue, never mind. So we've got this situation on the right-hand side of this semi-permeable membrane. Now, this is kind of the really critical point I want to make to you. Over on this right-hand side, we would refer to this as a concentrated solution. Okay, so it's concentrated. In other words, it's a solution. It's a mixture of sugar and water. but it's more concentrated. There's more of the sugar on this side of the semi-permeable membrane than on the dilute solution side, right? We can see that there's less sugar there. In fact, there's no sugar on that left hand side. Let me bung one in just to be fair. So let's bung one in there just to let those guys feel part of the party. So what we're going to find here is then that we're going to get a movement of fluid. Now the other point we want to understand, I think it's really important, is that these sugar molecules, they cannot fit through, if one of these guys tries to kind of go up this way, it's not going to fit through that membrane. Okay, it's not going to get through there. But of course, these water molecules can do exactly that. This guy here, for example, can through its random kind of kinetic movement, can jump across and pass through the membrane in that direction. Probabilistically speaking, it can happen. Now the question is, on this concentrated solution side, what is the chance of, say, this guy getting out there? Well, can you see immediately that the chance is lower of fluid from our concentrated solution getting out of the cell? Well, why is that, James? Why are you saying it's lower? Well, first of all, this guy here in the concentrated solution is potentially, probabilistically, in the way. There's more probability that our molecules from the left-hand side will pass across to the right-hand side than there are of the molecules from the right-hand side pass across the left. Some may well do exactly that, okay? But more, probabilistically speaking, will pass from the left-hand side to the right-hand side. Now, we can't describe that as diffusion because it's not simply an equalization of the concentration gradient. That water is not passing down the concentration gradient, but we are achieving what we would refer to as a net movement. Okay, we are achieving what's called a net movement. Okay, if these sugars weren't there, this would equalize and stabilize with equal pressure on both sides. But that's what our diffusion tells us is going to be the case. But the point we want to make is that over on this side, we have what we call a hypertonic solution. A hypertonic solution. In other words, hyper means that there's more of it. Okay, there's this sugar in here. And here we have, on this side, we have what's called a hypotonic solution. So the real critical point here is that generally speaking, we're going to get a net movement to the right onto the inside of the cell from those water molecules because they have probabilistically a greater chance of passing through than the water molecules on the right moving to the left, because that concentrated solution is kind of getting in the way. It's preventing it from happening in the same quantity. So this is a super important point coming up. As a result of this can actually cause an animal cell to swell, okay, and then, or even, and this would be a more extreme case, of course, what would happen if it swelled enough, or even burst. Okay, so this process, which we are going to describe as osmosis, actually explains how water will continue to move from one side of the membrane to the other as part of a dilute solution into a more concentrated solution. And it seems counterintuitive, but the reason it happens is because the solute, in this case, the sugar, kind of is partially preventing the water on its side of the membrane from escaping, from getting out, just the probability of it. And it can cause this animal cell to swell or even burst. And of course, we just want to note there, this is one of the reasons that plant cells have cellulose. Okay, we talked about that a few tutorials ago. The outer layer of a plant cell, the cell wall, contains cellulose. It actually prevents this osmosis from taking place. Okay, a couple of other points before we finish this off. Point number one, I'm actually going to call it case number one. Okay, so case one, let's see how this would actually affect things. Let's draw some kind of simple container and let's give it a little level there. And let's say that in this container we have pure water. Okay, pure water. So of course that is what we would call a dilute. Okay, it's not a concentrated solution, it's pure water. But let's also assume that inside that water we've got some kind of animal cell. Okay, so again we've got the nucleus in there. Maybe we can see some mitochondria. But the point is that cell is in there. And what we're going to find in this situation is, as that cell is in that position, because inside the cell, effectively, we're in this case. Inside the cell, effectively, we're in this case. So what will happen is this pure water from the external environment will start to move into the cell via the process of osmosis like this, and the cell will swell up. Okay, so we need to write here the cell expands. Cell expands, and we can say here it can burst. Okay, let's look at case 2. Just drag that down, bring that up a little bit. Let's look at case 2. Case 2. Okay, so case 2, exactly the same situation, but this time we don't have the dilute, which is pure water. This time, let's say for argument's sake, we have concentrated, let's argue that this is concentrated sugar solution. Concentrated sugar solution in there. Okay, so that's what we've got in there. And again, let's do the same thing. Let's put some, it's meant to look the same, it's not going to, you get the idea. Let's put some animal cell in there. Let's have a nucleus, shade it in a little bit, that dark spot there. Let's put a couple of mitochondria in. It doesn't really matter for now. Let's put those in there. And of course, what's going to happen in this situation is now we've got a concentrated sugar solution on the outside environment of the cell, and the cell, the inside of the cell now is the dilute. So what's going to happen through the process of osmosis? The cell is going to emit its water, or at least some of its water, and it's going to lead to what we'd call cell shrinkage. The cell effectively shrinks, okay? And we can describe this as a process of dehydration. So if we run for a long time, for example, we may become dehydrated, and this is the exact situation which is actually going to occur. And this is why, if we sort of put it into the context of something like sports drinks, okay, sports drinks, what we're going to find within those sports drinks is we're going to have a couple of things. Well, let me actually make 3 arrows here. Those sports drinks are going to have water, okay, to replace that which has been lost in sweat. They're also going to contain ions, minerals, things like sodium, for example. Sometimes we call them electrolytes. And it's also going to incorporate things like sugars, okay, which have been used up in respiration. And the reason that is to prevent this case from happening, to prevent dehydration happening by making sure that we maintain as close as possible to an equilibrium state so that the cell structure can remain intact, and osmosis is not going to ultimately shrink down that cell into like a shriveled nothingness. Equally, on the other side, the cell, in the other case, of course, the cell can expand through osmosis, as we mentioned already. That's all.